Lake Amatitlán used to be a very attractive lake for internally and externally tourism. As one of the most visited tourist sites in Guatemala, it created a great demand in the tourist industry, which generated a strong employment sector for Guatemalans and boosted the textile industry. Many people were able to sell their textiles, which were highly covered for being brightly colored and handmade. But in the recent years, however, things have taken a drastic turn. Currently, Lake Amatitlán is at risk of becoming a swamp, as it is exposed to constant pollution, which has made it difficult to find a strategy for rescue it on revenge to tourist attraction. Due to the decline of tourists in the lake, many people who work in the sector and the textile industry have been very affected. The lake is directly and negatively affected by the accelerated growth of the city and nearby towns. The consumption of forests and industry developed in the lake's watershed area. This has generated a process of accelerated eutrophication, which interrupts an excess supply of inorganic nutrients in aquatic ecosystems, mainly nitrogen and phosphorus, causing an uncontrolled proliferation of algae and bacteria. This has generated the following economic consequences.
We are the biotechnological research team of Universidad del Vale de Guatemala, Iktan Kaan Ek. We accompany and support us to be a community of great impact for the development of this new research. To promote tourism in the lake, we propose to modify the cyanobacteria Anabaena sp. They will be used to treat the lake so that they produce bioluminescence at night while cleaning the lake. The cyanobacteria will grow in the transparent flow reactor that will be in the shore of the lake. So visitors from all over the world can come to see how the microorganisms that turn green the lake also can clean it. And they will see how they produce natural light in a special and visual attraction at night.
Deciding what we wanted to do for our project took time and a lot of reviewing of new ideas. Our first step was to list some of the country's problems such as contamination, a lack of alternatives for sustainable materials, unequal access to water and energy. Synthetic biology was the starting point for solutions to the proposed problems. For that point, innovative ideas such as biomaterial creation, bioremedial strategies, alternative energies were proposed. After conducting research, eutrophication was selected, and reactivating tourism in Amatitlán Lake was the problem to be solved. Consequently, the team began investigation about water treatment based on genetically modified cyanobacteria. Anabaena sp. was selected as the organism that assimilate nutrients and reduces their concentration in the lake. Plasmid P-CAT000 was used to express genes bonded to luminescence production.
The isolation of the cyanobacterium Anabaena sp. is crucial for the correct transformation of these microorganisms and for the process proposed. The protocol proposed to achieve this consists of taking a sample of the water of the Lake Amatitlán in Guatemala with a plankton net in order to isolate the cyanobacteria present in the lake. Then, the sample has to be inoculated in a selective medium, like C8, without nitrogen but with sodium chloride. After almost 2 weeks, we have to isolate the filaments individually and repeat the inoculation in the C8 medium until obtain a purified characteristic culture of Anabaena sp. The purified culture has to be analyzed to corroborate that the microorganisms were correctly isolated. To achieve this, we decided to amplify the 16S ribosomal RNA gene of the Anabaena sp. because its sequence is highly conserved among different bacterial species. The result of the sequencing has to be analyzed in blasts of NCBI to corroborate that isolation of the cyanobacteria corresponds to Anabaena sp. For our design, we decided to use the plasmid pCat.000 as the vector for our project. This plasmid includes a replication origin for a broad range of hosts of cyanobacteria, and for that includes the RSF1010 Rep-A and Rep-B proteins that are necessary for the replication, and it has been demonstrated that works in Anabaena sp. To build our plasmid, we propose to use the Gibson assembly protocol because the construction of our plasmid requires the cloning of multiple sequences, such as the PSBA2 promoter, expression cassettes, BBAK, 216017, BBA K743015, and T7 terminator. These sequences can be obtained via PCR reaction with the properly constructed primers, which will allow us to incorporate the overlapping ends to each fragment for the insertion of the coding sequences into the plasmid. The parts added to the vector were the sequences that code for the luciferase, which is the enzyme that oxidizes a substrate to produce light, and the enzymes that produce the luciferin, which is the substrate oxidized by the luciferase. They will allow the cyanobacteria to produce bioluminescence. A part that includes a promoter regulated by the circadian cycle was used for the expression of the luciferase, and the PSBA2 promoter was used for the expression of the enzymes that produce the luciferin. To optimize our design, we decided to use the part with the regulation of the circadian cycle, so that the bioluminescence is only expressed at night, when it can be seen by people and the metabolic load for the cyanobacteria is reduced. These fragments are to be mixed with the Gibson assembly master mix. After going through the master mix reaction, a larger fragment composed of the annealed single fragments added should be formed, which should be analyzed too by a gel electrophoresis. This new larger fragment is the insert that is cloned to the pCat.000 plasmid, also by a Gibson assembly. We kept in mind that this protein expression cassette was added to the multiple cloning site of the mentioned plasmid, in the EcoR1 restriction site. Therefore, the primers for both ends of the protein expression sequence were constructed based on the left and right arm of the digested vector, contemplating the overlapping region sequences in order to allow the annealing of the inserts into the plasmid. Both the expression cassette fragments and the digested vector go through the same master mix reaction, previously described, in order to generate the transformed pCat.000 plasmid that will allow the proper expression of the luciferin and the luciferase. A verification of correct construction of the plasmid is recommended via gel electrophoresis or preferably through a DNA sequencing process. After having our vector ready, we propose to introduce it in an isolated native strain of Anabaena sp of the lake. Following the isolation of the cyanobacterium Anabaena sp, it was decided to perform electroporation for the introduction of the plasmid into the cells. The application of electricity induces the formation of temporary pores in the plasma membrane, and the electrical potential across the membrane causes charged molecules such as RNA or plasmids to enter the cytoplasm through the pores. In the electroporator cuvette, the prepared cyanobacteria are added together with the DNA plasmid, where 300 volts are supplied for 2 milliseconds to obtain competent salts. The plasmid includes sequences for kanamycin resistance, which will be helpful to select the transformed bacteria, along with the blue-white screening method. Blue-white screening is a rapid and efficient technique for the identification of recombinant bacteria. The blue and white colonies appear on the surface of the agar. The recombinant cells from the white colonies are selected for cultivation. On the other hand, we propose to grow the cyanobacteria in an isolated bioreactor, so that it consumes the excess nutrients in the lake while produces bioluminescence at night as a tourist attraction. The demonstration reactor for tourists connects to a main pipeline that transports the samples to a scale-up system of the extraction and purification of pigments methodologies. The system starts with the centrifugation of the flow, with the biomass sample. Inside a continuous decanter centrifuge to obtain sample pellets packing with pigments, which is the solid outlet. The liquid outlet is recirculated back to the lake after passing through the pretreatment process of membrane filtration with ozone and UV disinfection stage to avoid any form of contamination with cyanobacteria or plasmids. To design the modified plasmid, the expression cassettes of the proteins were introduced in the multiple cloning site of the plasmid, specifically in the restriction of EcoR1. The parts introduced in the vector were the part BBA K743015, which includes the LuxA and LuxB genes from Vibrio fischeri that code for the heterodimeric luciferase. Both subunits must be synthesized to have a functional enzyme. The part includes an RBS for each protein, and they both are regulated by the PTA BBA K743003 promoter, which is a promoter regulated by the circadian cycle, so that the enzyme is expressed after dusk, and just during the night, the cyanobacteria will emit bioluminescence, which is the time when it can be noticed and the bacteria does not waste resources in vain. It was verified that the coding regions have a stop codon, and after the last stop codon, the T7 terminator was added to end the transcription. Then, for the other expression cassette, the promoter PSBA2 from anabaena was introduced because it was considered to be a constitutive promoter by being highly inductible underlying, since it promotes the transcription of the gene that codes for the D1 protein in the photosystem 2. We designed the plasmid so the cyanobacteria produces the luciferin during the day, in which the bacteria makes photosynthesis to take advantage of the anabolism of it. After the promoter, the part BBA case 216017 was added, which includes the LuxC, LuxD, and LuxE genes from Xenohabdus luminescens that code for the reductase, a transferase, and a synthase respectively, which are necessary to transform the myristoyl ACP from the metabolism of fatty acids to myristoylaldehyde, which is the luciferin that is oxidized by the luciferase with oxygen and FMNH2 to produce water, FMN, myristic acid that can turn again to myristoylaldehyde and light. The part includes an RBS for each protein and a stop codon. Finally, the T7 terminator was added to end the transcription. The system design allows to produce the unaccumulated myristoylaldehyde, which is the luciferin, during the day, in which the cyanobacteria produces molecules that provide it energy, and at night it starts the production of the luciferase, which will have enough substrate and energy, ATP, to produce a considerable intensity of bioluminescence. In order to prove that our design was successful, we decided to verify that the genes in our plasmid would be expressed. For this, after modification of the pcat.000, a sequence map was obtained. This provides us all the generic information about the plasmid, which is an exportable as a FASTA file. To validate the expression of the gene and how it works, a bioinformatics software for the identification of genes, functional signals, and deciphers of gene expression, data called Softberry, was implemented. The advantage of this algorithm is that it works for genetic predictions, which provides an automatic annotation package for the prediction of promoters, terminators, and halterers. Also, this algorithm works with the automatic annotation of genes predicted by homology with COG and NR databases. All predicted genes showed that the synthesis of this according to the entered plasmid functionality, providing that our design works and that it can go to the build and test phase.
To test the results, we propose to verify the plasmid expression in cyanobacteria by measuring the intensity of the bioluminescence and the efficiency of the system by measuring the concentration of nutrients at the exit of the reactor. We also propose to collect the final biomass and extract pigment from it. We chose a plug flow reactor for our design. To determine the size of the PFR reactor in which the cyanobacteria will grow, it is necessary to determine the kinetics of growth with a monod model, in which nitrates will be the substrate of interest to be consumed since it is the main pollutant that causes eutrophication, and will be the limited nutrient since carbon dioxide will be supplied in excess. After applying the mathematical procedure, the volume of the reactor will be equal to space-time multiplied by the flow to be treated. The river that carries the largest amount of nitrates to the lake is the Villalobos River, which has a concentration in its effluent of 0.406 milligrams per liter and has a flow of 1.73 cubic meters per second. To reduce the concentration of this nutrient in the lake, the proposed system must reduce the entry of nutrients into the lake. So it is proposed to place the plant in the Playa de Oro Bay, which is near the Villalobos River outlet to prevent a large quantity of these nutrients to reach the rest of the lake. It is proposed to treat 25% of the river flow to have a nitrate conversion of 70%. To estimate the size of the system that the cyanobacteria will travel, a simulation of the reactor was carried out with the software Berkeley Madonna. For the simulation, we used the next kinetic parameters for nitrate consumption of the unmodified cyanobacteria. The simulation was done using differential equations presented that describe the growth of the cyanobacteria and the consumption of nitrates. For the conversion of the 70% of nitrates, the final concentration will be 0.123 milligrams per liter, and at that time, the concentration of biomass will be 951.9 milligrams per liter. We determine real bioreactor volume using differential equations, and we consider the flow of air that will provide the oxygen and carbon dioxide to the cyanobacteria for its growth, and the production of bioluminescence. Using an airflow of 5% of the flow of the medium, the real volume in the PFR will be 3,236.4 cubic meters per day, and the size of the reactor will be 3,186.8 cubic meters.
This is the model of what our bioreactor will look around the lake, allowing people to see luminescence at night.
Iktan Kaan Ek will show outcast itself by direct means with potential clients, preferably in the areas near the treatment plant, infrastructure near the lake. We provide personalized attention through Iktan Kaan Ek social networks as well as the telephone. The client enjoys a personal experience from the initial attention until he receives the product. This initial attention involves defining the characteristics of the that you want, as well as the quantity and the days times of delivery to your establishment. Our key activities include development of infrastructure on the shores of the lake, advertising and exposure on social networks, workshops in nearby communities to promote the product of sale, the dye, development of direct distribution network to target customers, customer support, and establish a relationship with the municipalities to study the contamination of the aquatic ecosystem in the question.
Application of synthetic biology were used to solve the problem of contamination in Lake Amatitan and thus avoid contamination by toxins produced by some species. However, the cyanobacteria species being used in a study has a BSL-1 safety level. By treating a low-risk species, basic safety protocols for the use of genetic material must be followed in the laboratory. Some of the protocols consist in having appropriate protective clothing, emergency equipment that includes at least one sink, one emergency shower, and one wash station, and material properly marked or identified in order to be discarded or properly sterilized. Before using them again, staff must be disinfected before entering and after leaving the laboratory to avoid contamination, and should record all the transgenic material used and must be discarded in appropriate and identified container for later incineration. All surfaces must be disinfected, and after finishing or when there has been a spill, to eliminate any microorganism or tracer of genetic material. And remember that the relevant tests must be carried out of cyanobacteria on a laboratory scale to ensure that they are safe for the environment, people, flora, and fauna.
Sharing our project with people all over the country was a main priority for us. We decided to share our project with different school students from various schools all over the country. The main objective was for students to understand the power that synthetic biology has to change things in little communities in our country. We conducted activities so that people of all ages could understand what our project was about. We also conducted virtual and in-person workshops, so if people were interested in synthetic biology, they could come and learn all about synthetic biology and its powerful applications to solve problems in little communities in our country.
We also considered it important to share ideas and knowledge with others around the world. For that, we shared time with Biotech EC and discussed about synthetic biology and our goals to positively impact society with the safe and innovation alternatives.
This is the second time that our Iktan Kaan Ek team participates in the IGEM Design League. And like last year, we are very proud to present our final project as a multidisciplinary team. Teamwork, diversity, innovation, commitment, and excellence were our main strengths that guided us throughout this experience. Likewise, synthetic biology represents an important component in our trajectory, since it allows us to explore the potential of biotechnology to solve problems in our country, Guatemala, in an innovative and population-focused way. On this occasion, we have shown the process we had to design a biological system based on the genetic modification of cyanobacteria, Anabaena sp, to reduce the levels of eutrophication in Lake Amatitan, while producing pigments of importance for the textile industry and producing bioluminescence to reactivate local tourism as an important source of economy for the population. This demonstrates that the pinnacle of the process allows the development of modules that can solve in a revolutionary and scalable way important problems based on the principles of synthetic biology. We close this stage with many learnings as a team, and above all, committed to continue with this trajectory in the following years.